Welcome, wherever and whenever you're watching this poster video. And well, already thanks the interest, and I will try to talk you through the most important points of my poster. So, the setting we have here is the following. Imagine we have a cancer patient, and we want to identify mutations in the tumor. And to this purpose, we take samples of the tumor and also of the reference germline of the patient, and then we sequence them both. And so there are automated tools which will look through the sequencing data and then compare between the tumor and the reference, and will flag positions where it alleges there might be a variant. The issue is, however, that very frequently misalignments or sequencing errors or other mistakes are flagged as a discrepancy, as a potential variant by these tools. And so if we want to get to an actual list of mutations at the end, what is the state of the art right now is that human researchers have to sit down and manually classify the candidate variants following standard procedure. And this is obviously both very time consuming and also not very reproducible, seeing how for some edge cases different people may have very different opinions. And so automation is the obvious solution to both problems. And so what we took on as a task is to provide a reliable way to automate this decision process. And one factor which plays into it, which is an important nuance here, is that besides the usual very well-known aspects of recognizing artifacts versus mutations, is that it's also necessary to consider other sequencing files which were made in the same library operation, which sounds odd. However, there are certain positions which generally look clean, but will always have a deviation from the reference throughout all samples of one library. And so when it then happens that the germline and the tumor were sequenced in different libraries, then we'll get a bunch of discrepancies which are sequencing artifacts, which are not real mutations, but which we can't recognize as such. So what we have to take into account is not just the two samples we have, the tumor-normal sample pair, however, also context samples. And so once we have that, we utilize a relatively straightforward approach from machine learning, which is convolutional neural networks. And so you probably have heard about them from other areas such as image recognition. And they generally excel whenever there is important spatial structure in the data. And in our case, the alignment of the reads and obviously the base sequences inside are very, very important for us. So after formulating the sequencing data into numerical form, we feed the data which was previously manually annotated through a relatively straightforward model architecture, where we basically, in the beginning, have convolution blocks with max pooling every time in order to learn high-level features in some sense. And then the result, the output, is fed into just a fully connected classification head. And this is a structure which is also frequently used in computer vision. And so as a result, we see that this performs quite well. So if we perform five-fold cross-validation, we get scores for both accuracy and precision recall F1, which are all in line with what one would expect from a qualified trained human following a published guide. And so this shows that indeed this approach can be used to reliably automate the distinction between mutations and sequencing artifacts. And so, thank you very much for your attention. If you have any questions, I will be very happy to answer them. Contact me in any way.